Okay, so a little quick review before we head on. So what's the difference between the humoral immune system and the cell-mediated immune system? What does humoral do that cell-mediated doesn't do? Well, it has to do with antigen presentation, but actually both cell-mediated and humoral do. But in humoral, when antigen are presented to B lymphocytes, they do something important. What do the B lymphocytes do? They produce antibody. Okay? So the whole goal of the humoral immune system is to activate these B lymphocytes to produce antibody. And what does antibody do?
Attacks the pathogens?
Yeah, I mean, you could say it attacks it, but it really kind of marks it for other things.
Attack.
Attack, okay.
Attack.
Gotcha.
That's what I'm saying.
It marks those pathogens for destruction by other parts of the immune system. So what does the cell-mediated system do if it doesn't produce antibody?
It kills them?
It kills cells that are infected or damaged in some way, be it cancerous or virus infected. Okay, so it's pretty important. What is a type of cell that is an antigen presenting cell? We've actually talked about two of them so far, and there actually are three. Macrophages are antigen-presenting cells. And there's another cell related to a macrophage, almost kind of a cousin, if you like. Dendritic cells, okay? Dendritic cells tend to be located more in the skin. Interestingly, we're going to see later on that B lymphocytes, the ones that produce antibody, can also be antigen-presenting cells. So that's another wrinkle we'll talk about later on. Okay, we've divided up the immune system into three groups or lines of defense. And two of those are considered to be innate, meaning that you are born with them. Which two? Line one, line two. Line three is one you develop during your lifetime. That's the line that happens a lot in the lymphatic system and involves this interaction between an antigen-presenting cell and a B lymphocyte or a T lymphocyte. That's what we call our adaptive immune system. And that's the one that really differs in every individual depending upon what your prior exposure has been. Some of you have been exposed to measles. Some of you have not. Therefore, some of you have immunological memory of being exposed to measles. Some of you do not. And that has consequences if you are ever exposed to the real thing. Okay, so what we're going to be doing is we're going to be going through those three lines of defense, spending most of our time on the third line of defense, that adapted line of defense, the cell-mediated and humoral immunity. But we also do need to understand lines 1 and lines 2 as well, because they are important. Line 1 is primarily your barriers of defense. And the goal there is to prevent infection. By definition, what has to have happened for infection to have occurred? You have to invade the tissues, and those organisms have to multiply. Does infection always cause symptoms? No. Remember, we can have some asymptomatic infections. If we have symptoms, what are they typically the result of? What causes the symptoms associated with tetanus, for example? Virulence factors that are produced by the pathogen, toxins, or exoenzymes. Okay? Now, additionally, symptoms can be produced by your immune system. We talked a little bit about interferons and how that part of your defense against the spread of infection can also result in some of the symptoms associated with particular diseases. So what we said was that the first line of defense is kind of like that old Western fort in a cowboy movie, a barrier against invasion. Okay? So we don't want the pathogens to cross that particular barrier, and so there are certain adaptations associated with the line of one of defense that tries to prevent infections from occurring in the first place. And we typically break those line one defenses down into 3 subgroupings, and they have to do with kind of physical barriers, chemical, and then genetic considerations. The major 2 physical barriers are skin and epithelial mucosa. And associated with these 2 barriers, there oftentimes are a variety of biochemical substances which kind of reinforce these protective barriers. And those are some of the things that are illustrated on this little handout right here, this diagram right here. For example, in both saliva and tears, we have a natural flushing action, particularly with tears, to wash pathogens away from the conjunctiva of the eye, for example. And if you get a lot of dust in your eye, what's one of the responses that happens? Your eyes tend to water more, to tend to kind of wash things away. So kind of the windshield wipers of your eyes, almost. But furthermore, built into that washer fluid are some biochemicals that are antibacterial. Very specifically, what's one of them? One of the major substances in tears and saliva. It's antibacterial. Lysozyme. Okay, lysozyme is that substance that degrades bacterial cell walls. What part of the skin is most notorious for getting infected? Where do you think there might be the greatest chance?
Hair follicles.
Hair follicles, pores in your skin, sweat glands. Okay? So in the hair follicles, we have some sebum that is produced. Sebum tends to have a little bit lower pH. It tends to have a little bit of antibacterial activity. Sebum also tends to keep the skin from getting too brittle and therefore for breaking and becoming compromised. Sweat itself has some antibacterial properties. Sweat has a lot of fatty acids. In addition to the normal outwashing that sweat provides in kind of moving things out of your pores and cleansing them, that lower pH that the fatty acids produce tends to be antibacterial in nature and to retard some microbial growth. Flushing of substances out of your body is a fairly normal strategy to try to prevent infection. So the process of urination, for example, helps to wash pathogens out of the urinary tract. Now, if they have fimbriae or pyeli and can adhere to the walls of the urinary tract, that flushing action isn't going to be quite as effective. What's a flushing action that's associated with your GI tract that tends to move pathogens out of your body? does your GI tract do to move things through? Peristalsis, remember that rhythmic contraction of your gut? And what is oftentimes a response to infection? An increase in the rate of peristalsis. And what does that do? That moves things through your body more rapidly. The less time that material spends in the colon, the less time there is for water reabsorption. And so increased peristalsis tends to lead to a more watery stool. So diarrhea is really many times a consequence of your body kicking in a defense mechanism and going, get this stuff out of here. So should there be some thought given, if you're suffering from diarrhea, of taking an anti-diarrheal like Imodium? Yeah, probably. Okay, because you're compromising then one of your body's normal defense mechanisms. Now, sometimes it's necessary, but sometimes you might be able to get by without it, and you probably shouldn't take it unless it's really, really important to do so. There are some biochemicals that are secreted into the large and small intestine that help to also protect against infection. Specifically, substances called bile salts. Where does bile come from? Pardon me?
From the liver and then gets
Stored in the gallbladder and then emptied into the digestive tract. What's the purpose of bile in digestion?
helps with the fat or something.
Yeah, it's an emulsifying agent. It helps to break up the fat into little droplets that are more easily digested. But bile salts, which are one of the components of bile, are also inhibitory to gram-positive bacteria. So they are an antibacterial agent. Before substances that you ingest get into the small and large intestine, they have to go through the stomach first. So basically, when you eat, you drop what you eat into a vat of acid. What's the purpose of that acid? Well, there is some digestive purpose, but it's fairly minimal. The major purpose of having that vatted acid at the end of your food tube is to drop the food in there and kill the bacteria that are on that food that you eat. There's another important substance as far as microbial growth is concerned that I mentioned earlier on, and that is iron. Iron can be a limiting factor to microbial growth. And if there's not enough iron around, it inhibits bacteria from growing. Iron has to be transported in our blood. And it's transported as being part of hemoglobin, obviously. But then there are also other proteins in our blood which also transport iron around the body, including transferrin. Iron's not very water soluble. It's not going to just dissolve in the blood fluid. It has to be bound by proteins in order to be transported. So this transferrin protein has a pretty high affinity for iron. Staphylococcus aureus has the ability to produce a protein of its own called syrtophores. And these actually have a stronger affinity for iron than blood transferrin does. So if Staph aureus starts infecting your body, particularly your blood, it can grab iron from your iron transporting proteins. So there might be kind of a tug of war for iron going on within your body, which will have a big impact on whether microorganisms can grow or not grow. Another strategy that pathogens have used is that instead of engaging in a tug of war for iron biochemically, they just lyse red blood cells. And so by producing those hemolysins, particularly the beta hemolysins that both lyse the red blood cell and break down the hemoglobin, they are able to increase their iron availability in that regard as well. Skin has protective cells associated with it, particularly the dendritic cells. You can also find dendritic cells in the epithelial mucosa. We mentioned earlier on that epithelial mucosa can also have cilia associated with it, and the sweeping action that ciliary escalator has helps to move pathogens back out of your respiratory tract, for example. So there are a number of different mechanical, biochemical, and physical barriers, which are listed in these two particular tables right here, that really help to make up the first line of defense and are really pretty effective in terms of preventing microbial infection in the first place. What about the genetic components of your defense system? One of the keys for genetic factors influencing your first line of defense have to do with receptors. Because remember, if infection's going to take place, the very first thing that has to happen is attachment. If things don't attach to the tissues, the infection's not going to occur. That attachment is usually highly specific and involves receptors on the surface of the tissue being complementary to molecules on the surface of the pathogen. TB is a good case in point. Tuberculosis had its origin as a disease in Europe. And for centuries, it ravaged Europe. But over time, what happened? People started to develop a natural immunity, a little bit, not completely, against the pathogen TB. What did that involve? Well, one of the things that it involved were minor changes in receptors so that the TB agent had a more difficult time attaching to cells. What did that involve? Mutation. What was this example of? The development of more resistance to tuberculosis in European populations was the result of natural selection. And individuals who had that mutation that were less likely to have the TB pathogen bind to their cells were more resistant, they had a greater chance of survival, and then they could pass those genes on to their offspring. Well, when Europeans colonized America, the Native Americans and the native populations had never been exposed to TB before. And so they were highly susceptible to the disease. They were also just very susceptible to another disease of European origin, smallpox. And so people, particularly carriers of that disease, people who were asymptomatic, brought over those pathogens, and the pathogens found a completely compromised new population of individuals which had very little natural resistance to tuberculosis. There's some pretty good evidence that during the height of the American Indian Wars, that one of the Army's strategies were to provide smallpox-laced blankets to Native American tribes carrying out bioterrorism. pretty dark period in our particular history. And if that indeed is true, that's pretty serious stuff. Another genetic consideration are that some genes are pleiotropic. Remember from genetics what pleiotropic means in terms of genes? Pleiotropic genes are genes that can affect more than one trait. The sickle cell gene is a good example of a pleiotropic gene. What is the heterozygote advantage of being heterozygous for the sickle cell gene? Resistance to malaria. Okay, so sometimes the evolution of genes gives you an interesting pattern of resistance. One of the most dramatic genetic considerations associated with disease is a condition called SCIDS. SCID stands for Severe Combined Immunodeficiency Syndrome. And babies who are born with this particular disease virtually have no immune system. It interferes with their ability to produce both B and T lymphocytes. Interestingly, SCIDS is the result of a single gene defect. One gene being wrong can compromise your whole immune system. The ADA gene, the adenosine deaminase gene, is the culprit in this particular syndrome. So if you're born with SCIDS, what are your potential outcomes? Not good, no immune system. Unless it is diagnosed pre-birth, and following birth, you are transferred into a sterile environment where you live your life. Avoid the plastic bubble. There have been some attempts to try to correct this particular gene defect through genetic engineering. And there have been some successes. However, several years ago, the last clinical trial I think that was done in dealing with SCIDS and trying to correct that gene defect was halted because they were using a virus vector to introduce the gene into the affected individual, and it dramatically increased the rate of leukemia, and so they suspended those particular trials. But think about it, a single gene defect basically knocking out your whole immune system, your entire immune system. That's pretty dramatic.
I have a question.
Yeah.
Is there like an organ or something that babies have that goes away over time that helps with immunity and everything?
Mm-hmm.
Would that be missing in this mutation?
That's a different syndrome, actually. That particular syndrome is called DiGeorge syndrome, and we'll talk about it. And the organ's the thymus. And so people have a defective thymus. That affects your T cells, but not your B cells. So it doesn't knock out your entire immune system. So we'll talk about DiGeorge syndrome in a little bit and what the thymus does and what it does. So what if your first barrier of defense is compromised? Well, then the second and later the third lines of defense become activated. Before we get into specifics about the second, third line, however, we need to do something else. And that is to try to give you a better appreciation for kind of the overall organization of your immune system. So let's look at the immune system from kind of the macro level first, the big picture. And once you're more comfortable with kind of how it's organized and what it consists of, we'll get back and dive into lines 2 and line 3 and see how it operates. Let's think of the immune system right now as a series of connected compartments. And these compartments that make up your immune system are pervasive, meaning they're all throughout the body. There's not one particular location where we say, this is where the immune system is. It's all over the place. So, big picture, the 4 compartments that are connected, that are pervasive throughout your body, are a system called the RES system. RES stands for reticuloendothelial. Reticuloendothelial system. The extracellular fluid is one of the compartments. This is what bathes the cells that make up your body's tissues. That's the tissue fluid, if you like. Your lymphatic system, and we'll spend a fair amount of time talking about the lymphatic system. And then your blood. These first 2 compartments we're not going to spend much time talking about. I think it's important to hear the terms. The reticuloendothelial system and the extracellular fluid. The RES system consists of a bunch of fibers that emanate from tissue cells, particularly connective tissue. And these fibers are called reticular fibers. The cells that produce these reticular fibers are bathed in this extracellular fluid. So, if you look at this diagram right up here, here's a representation of some RES tissue. And here is a bunch of connective tissue cells. And they produce all these little reticular fibers that kind of fill the spaces in between the individual cells. And then this space is also filled with that extracellular fluid. Over at the right, it shows you places in the body where there are high densities of this RES tissue, where there are high density of connective tissues. Now, what does this have to do with immunity? Well, populating this RES compartment are a variety of immune cells. Particularly macrophages and neutrophils. Macrophages, and neutrophils are produced by the bone marrow and then are released into the blood. So how do they get from the blood into this RES system? They actually crawl out of the blood vessels and into the surrounding tissues by a process called diapedesis. Do any of you remember from cell biology, you might have seen it in other classes as well, seeing that animation, the inner life of the cell? And one of the opening shots of that is that you've got these white blood cells rolling through the blood vessels and then grabbing onto a blood vessel wall and then squeezing in between the vessel cells and into the surrounding tissue. That particular process is the process of diapedesis. It's also called leukocyte extravasation, fancy word for crawling out of the blood vessel and into the surrounding tissues. These macrophages and neutrophils, once they're in the RES system, can either kind of settle down and become fixed in a particular location or continue to wander around. And if they wander, they tend to wander along those reticular fibers, kind of like train tracks. If they become fixed, they tend to become attached to those reticular fibers and localized in a particular part of the body, like in the chest cavity, where there's a potential for entrance of pathogens, or the gut. And what do these guys do? Phagocytosis. This is your first line of defense against infection. And if you are a macrophage and you phagocytize something, what else can you do? You can display those antigens. Once you've displayed your antigens, if you then migrate to the lymphatic system, you can show them to B and T lymphocytes and help to activate that third line of defense against infection. So this shows you a little bit of the RES system, and particularly the relationship between the connective tissue with its reticular fibers, the protective white blood cells, and then also the relationship to the other two compartments of your immune system, blood vessels and lymphatic vessels. So if something gets into the RES system, it can also enter the lymphatic system. It can also enter the circulatory system. And so all of these particular compartments of the immune system, the extracellular fluid, the blood, the RES, and the lymph, are all interconnected with one another. So an infection in one place can result in a response in another place. And that's important. Okay, let's spend some time on the lymphatic system. What are the roles of your lymphatic system? What's its purpose? Drainage, good. To return tissue fluid back to the circulatory system. To correct edema. Edema is swelling. So if some of your tissue becomes swollen, let's say you have an insect bite or you have an injury, does it stay swollen forever? No. Why did it swell in the first place? Because fluid left the blood and moved into the surrounding tissue fluid. That we'll see later on is one of the responses or outcomes of inflammation. So inflamed tissue becomes swollen. However, lymphatic vessels eventually reabsorb that tissue fluid, and the lymphatic system opens up into the circulatory system. Here's the right subclavian vein. Here's various lymphatic ducts opening into there. And so that fluid that was originally lost from the blood and accumulated in the tissue is now returned back to the blood again. That's really important because you need to maintain normal blood volume. And if you don't, you can go into hypovolemic shock and you can die. So swelling, if it isn't resolved, can be life-threatening. That's one role of the lymphatic system. What's another role? What's our contextual role? Now, this is where a lot of the immune interactions take place. So it's a major site of immune interactions. This is where pieces of phagocytized pathogens are shown to be in T lymphocytes. And then they're activated. And then cell-mediated immunity and humoral immunity is kicked into play. What's the third physiological role of the immune system? There's another role. It's nothing to do with immunity. It's nothing to do with edema. Do you recognize that term, lacteals? Lacteals are part of your lymphatic system. They're in your gut. And they are specialized to absorb one of the products of digestion. What are they? What's absorbed into the lymphatic system as opposed to the circulatory system? Has anyone ever seen lymphatic fluid? Know what it looks like? It's yellowish white, and it's very glistening. What gives it that glistening appearance? Fats. Fats are absorbed into the lacteals of the lymphatic system. They are not absorbed directly into the circulatory system. So that lymphatic fluid has a lot of fats in it. So, what are the major components of the lymphatic system, and where's it located? Well, it's located all throughout the body. Probably the part of the lymphatic system you're most familiar with are the lymph nodes. And you find lymph nodes concentrated in a number of locations in the body. Where do you find lymph nodes a lot? Where would you palpate lymph nodes to check for infection? Neck is one place. Where else? Armpits, you find a lot of lymph nodes there. You find a number in the abdomen as well. We'll come back and talk about nodes a little bit later on. There are also lymphatic vessels, and they permeate all throughout your body. And they are oftentimes in close association with your major vessels of your circulatory system. And this diagram over here shows you the location of some of the major lymphatic vessels. But they have to be all throughout your body, because they have to be able to reabsorb that tissue fluid and return it back to the circulatory system. Technically speaking, bone marrow is part of the lymphatic system. The thymus is part of your lymphatic system. Bone marrow and thymus are oftentimes referred to as being primary lymphoid tissue. Lymph nodes are considered to be secondary lymphoid tissue. As are your spleen, your appendix, your tonsils, your malt, and your galt. These are all considered to be secondary lymphoid tissues as well. Some of those structures are going to be unfamiliar to you, so we'll have to go through those a little bit. Let's mention primary lymphoid tissue first. One of the really important things that the bone marrow does in its role as a primary lymphoid tissue is it produces B lymphocytes. Once those B lymphocytes are produced, those B lymphocytes become educated. This process of education involves those cells developing, or probably building is a better word, a specific receptor for a specific antigen. So part of this process of B lymphocyte development is producing a cell that is a particular receptor that matches a particular antigen. Lymphocytes can be produced and are produced that match both self and foreign antigens. Is that problematic? Yeah, because if those cells were allowed to survive, we could develop autoimmune disease. Autoimmune disease is serious, oftentimes lethal. So what's another event that happens within the bone marrow? These cells are produced, these that respond to self are eliminated. And only the ones, at least theoretically, that respond to foreign are allowed to survive. These educated cells that survive are primed for action, but they're inactive at this point in time. What would activate them? Exposure to their matching antigen, the one they have the proper receptor for. How are they exposed to that antigen? By an antigen-presenting cell, one that had gobbled it up, displayed it, and then was showing it to a particular B lymphocyte. And only if everything pairs up properly is that cell activated, and then it starts dividing and doing things. Starts acting. Starts producing antibody if it's a B lymphocyte. So these B lymphocytes have an interesting history. They're born in the bone marrow. They gain their knowledge about interacting with a particular receptor associated with a particular antigen. But once they've got that knowledge, they just sit there. So as soon as they finish school, they go on vacation. And they might be on vacation their whole life. If they never encounter their antigen, they just always sit there in that state of dormancy with their feet propped up. But if they are exposed to their particular antigen, then they go to work. And they start dividing and doing things. So they're born in the bone marrow. They're educated in the bone marrow. But once they are educated, then they move out into some of these lymphatic tissues, like the spleen or the appendix or the tonsils or the nodes. And that's where they take their vacation, and that's where they wait for encounter. Arthur, did you have a question?
So you said they're producing their receptors, and then they wait, and then we get infected with an antigen, and gets, it works.
That
out.
binds
So-
to the receptor, and then that turns the cell on to start producing antibodies.
So does that mean there's only like so many receptors there can be?
Okay, so that's really going to be an interesting part of this discussion is how do all these different kinds of receptors get built? Because every cell has a different receptor on its surface. How many different foreign antigens are there out there potentially? Billions. So as it turns out, this process of education results in billions of different kinds of cells, each with its own receptor. How does that happen genetically? That's something we'll talk about. Okay? Out of time.